Hello, Pooja ma'am. I hope you have access to share the screen.
Yes.
Okay, so I welcome you all in our master class on microbiome and GI disorders. And today we have with us Dr. Pooja BM. Dr. Pooja holds a doctorate in gastrorenal system, and she will be our guide for today's master class. Also, we have a good population in the class, so to avoid in-between mic disturbance, we are keeping you all on the mute, and afterwards we can have a doubt clearing session. Also, one more important announcement after the class, please do not leave the group. So by tomorrow afternoon or max by evening, we will share the certificates with you all in the group. So kindly be in the group after the class also. Over to you, Pooja ma'am.
Thank you, Siddesh. I'll just share my screen.
Sure, ma'am.
Okay. I hope my screen is visible to everyone.
It is, ma'am.
Okay. I welcome you all to this course on microbiome and gastrointestinal disorders. Okay. So, in this session, you're going to study about the diversity of the gut microbe. Okay. We are focusing on the gut. Okay. As well as the disorders that are prone because of this alterations in the gut microbe system. Okay. So, this why we have to study the gut microbe and the gastrointestinal disorders. Okay. So, gut microbe is like the next generation medicine. It is one of the part that need to be learned when you deal with the next generation medicine, because it is a it is a main, it's one of the common target in the future. Okay, because in the future we will target the gut microbe, because you know that how much we are us is more than how much microorganism is present in us. Okay, we might have few number of cells, but the microbes, the population of microbes in our system is more, or 100 times, or 1,000 times than us. So this is why we it is important to study the gut microbe. Okay, because it is it is like what it is a modern medicine. Okay, which is again coming back to the old ideas, because in the previous, in the olden days, it is said that food is a medicine, right? So again, learning gut microbe also, it has a wide area of gut microbe is not just about gastroenterology, or it's not just about microbiology. It has a diverse fields. It can be, see, again I told microbiology, all this, the study of the microorganisms, the gastroenterology where you can study what are the disorders happens, how to treat, also about manufacturers of the specific treatment for this, using probiotics, the specific drugs, targeted therapies, also about the next generation treatment or diagnosis. Also, here the nutrition diet also plays into the picture of the gut, in the gut microbe, as well as the gastrointestinal system. Okay, this is better for me. So I will start my session by saying bacteria can make us healthy and keep us healthy. Okay, so bacteria, the healthy bacteria make us healthy, as well as the other pathogens make our system unhealthy or causes several diseases. Okay, bacteria have co-evolved with us and have been serving since the time. Okay, it has come since a long time, since the the beginning of the system. So they are the silent partners of good health. They optimize the power of our immune system to fight diseases. So if you see here, the gut microbe, right, the gut microbe, it is very complex. So it is like a consortium. It's like a consortium of several bacteria, fungi, viruses, archaeas, and protozoans. Okay. So the assemblage of all these microbes and their genome and the environmental factors. So this completely form the gut microbiome. Okay. I hope it is clear what the gut microbe is. The microbiome, their genetics, their genes, as well as the environmental factors. Okay. So the microbiota in the healthy gut. So like I mentioned, we are not alone. So we carry trillions of microorganisms with us, which I told you, which is a diverse, which is very diverse. It consists of bacteria, virus, fungi, protozoans, all this. Okay. But the gut microbiota is mainly, it's strictly composed of anaerobes. Okay. And the main two groups, okay, there are wide varieties of microbiomes that is present in the intestinal, in the gut. Okay. uh, there are more than 50 bacterial phyla that is detected in the human gut. Okay. So in them, the main, the important, the dominated two phyla are, the main dominated phyla are bacteroidetes and firmicutes. So these are the dominated phyla of the microorganisms that is present in the healthy gut. Okay. So like I mentioned, it is very diverse, and this is some of the common, the dominated phyla that I've mentioned here. So this microorganism, right, it varies. Okay. If you see our digestive system, you can see from the, from mouth to the anus, the our, digestive system, that it follows like that. From there to the anus, our the microorganisms, the diversity changes, the numbers also changes, the numbers as well as the diversity changes. Okay. So usually the microorganisms, they are very high in the oral cavity. Okay, we know that it is very high in the oral cavity, right? It's very high in the oral cavity. So mainly all this proteobacteria or firmicutes. Okay, the, like I mentioned, the common groups. Okay, that is present here. So in the oral cavity, they are very high. Next follows is the esophagus. Even here, the microorganism numbers are quite high, and they have a high number of firmicutes. Okay. Next, if you see the stomach, in the stomach, you know that there is a secretion of HCl, because of which what happens? It is acidic, right? Because of this, what happens? The microorganism level decreases. Okay. Even though it is decreased, there are few microorganisms present even here, but the number will be decreased compared to the oral cavity as well as the esophagus. Fine. Next, if you see this, the gut, the large and the small intestine. So in the small intestine, in the small intestine, in the duodenum, okay, it is relatively low. Okay, it is relatively low. Okay, because of some of the acidic environment present here. Okay, but they are few microorganisms present here. Next is jejunum. Even here, the population is quite low. Okay, and here the majority is bifidobacterium is present. Next, in the ileum, it is increasing. Okay, so you can see here, it was more, it decreased, and it started increasing in the ileum. Okay, it's slightly higher compared to all this. Next, the large intestine. Okay. So large intestine is it is like an hub. So here majority of the microorganisms are present. So the in the the large intestine, cecum, it has a better, the more substantial micro, or the community is present here. It has the drivers of bacterias, like firmicutes, all these are present. Next, the important components where the lot and the diverse and the good amount of the microorganisms is present in the colon. So in the colon, so in the various part of the colon, we can see you can see the rich and the good microbes that is present. The good communities of microbes are present. So when I say rich, it will be usually bifidobacterium, clostridium, all this, the rich microbe or lactobacillus, all these are present here, okay, in the colon. So if I say anything is happening, the gut, so if any alteration is happening in the colon, so that will result in the disease, okay, and the disorders that we are going to study. I hope you're clear with this. So you can see how the population as well as the diversity of the microorganisms, the varies along the different stages, okay. Next, you can see here some of the major bacterial phyla. Like I mentioned, these two are the major bacterial phyla that is present in our human gut, right? So you can see here what are the species it is present. You can see I mentioned, right, when I say rich, good, okay, it is lactobacillus, clostridium, enterococcus, ruminococcus, all this, bacteroids, glanibacter, bifidobacter, all these are the good bacteria. These are these are helping in the healthy system. If a person is healthy, it means he has all these bacteria in them, okay. So you can see how the arrangement it is there. So the first, this will be almost about 60 to 80% will be present here, and here about 15 to 25% acinetobacter. Acinetobacter is very bad. This is acetinobacter. See, there's a difference between acinetobacter, okay. This is actinobacteria. So the actinobacteria is about 2.5% to 5%. Proteobacteria about 1 to 10%. And this is about 0.1 to 2.2%. So this is the arrangement that is present in the healthy gut, in the healthy human gut. So I hope you're all clear with the basics, okay. The microbes, we studied what are the microbes, the gut microbes, what is present in the gut microbes, how the arrangement is done. Now let's see, where does this microbes come from, okay. So like I mentioned, the microbes comes from the birth. So I told the microbes, it evolved with us, right? So it came from the birth, okay, through seeding and feeding, like from birth as well as the feeding. What are the food we are taking, okay, from the birth as well as from the feeding what we are doing. So you can see here in the birth, so the the diversity as well as the composition of microbes, it varies with a vaginal delivery as well as C-section. So the variation happens. Why a human, sometimes if the mood of a person is, if I say the stress, the if any stress comes in a person, if any action or the anxiety level, everything is related to the microbes, okay. How the person is interacting, to any kind of problems, that also depends. The studies have shown that that is also depends upon the gut microbe composition that is present in that human, okay. So that is, the one person is different from the other person. The response to a particular kind of any stimuli also varies, right? So that is because how they have adapted the microbes, means how their microbial diversity is formed. So it starts with the birth, where it's, sometimes it is vaginal or C-sections. So there is a difference, right? Next, when the infant, uh, below the age of 1 year, the milk consumption, the solid food. So what the food is consumed, what is the milk product, either it is a breast milk or any other composition. If you're feeding the kid, the gut microbial diversity also changes. The toddler, again, the adult microbes, the full diet, whatever is he's eating based on their you know based on their tradition or anything, because uh, with the region, with the different region, people eat different kind of food, right? So with that, there, the gut microbe system varies. Okay, so if you see the the mode of the childbirth, I told you it is has a very significant impact. Okay, so it is said that the vaginal delivery, right? The vaginal delivery, no, the baby that comes from a vaginal delivery, it carries a good amount of microbes in them. Okay, they carries bacteroids, bifidobacter, bifidobacteria in them. Okay, so because of this become the first colonizer. Because see, this is how it started. Two persons will be like, if if I take a two individual, so you can see the vaginal delivery and the C-section. So he already is carrying the bifidobacterium, bacteroids, because of the vaginal. But here in the C-section, it is very less. The microbes is very less. There will be usually not present of the microbes. Next, also from here, the milk consumption. So the milk consumption, again, it varies about what kind of the food. Maybe the breast milk or any formula milk. Sometimes they will not have a breast milk, so they take some kind of formulas, right? So based on that also, your microbe, the microbial components in your gut will varies. Okay, so bifidobacterium, lactobacillites, they provide good colonizing you know factors. They are good factors, they are present in the breast milks. Okay, but here in the formula fed infants, okay, they will have a streptococcus mainly. The studies have shown that they will have enterococcus, clostridium, streptococcus. You can see here. So in the breastfed baby will have, mainly they will have bifidobacterium, lactobacillus, and the formula fed, they have usually streptococcus, enterobacteria, enterococcus, anaerobic streptococcus will be present. Next, the diet. So the full diet. So based on the diverse diet, food, whatever they are taking, their gut microbes, the varies. So they will start adapting to the food what they are taking. So their digestive, the microbes that is present in the gut changes. Okay, so usually in children's between 1 and 7 years of age, there will be great number of enterobacteria, enterobacteria than the adult. Okay, then the composition will varies. So it goes on varies. So this is about the formations. Okay, this is how the microbes have evolved, have it is come, how it is fed, that you can see here. Okay, so devoted to the development of the infant microbe infrastructure. So like I mentioned, it started prenatal also, during the pregnancy time, the breastfeeding, the solid food instruction, introduction and transition to the family diet. So immune system development is crucial as an infant moves from the protected environment in the womb to where the immune system has to act, adapt, and resultant. So what happens in the prenatal stage? It will be present. It will be protected in the womb. So as the child grows, the postnatal or the infant, the child in the childhood, these microbes, okay, they vary. So there is some kind of reaction happens because of the adaptations. There will be the act and adapt, okay, there will be productions of certain immune systems, okay. So what I'm going to say here is in this from the slide. So microbiomes are also has a function with the immune system. So if your microbes, if you carry a good microbiome, okay, good gut microbiome, a person will have a healthy immune system as well. Okay, that we will see the process how the immune, the gut microbe as well as the immune system, how the variation happens. So this is what I mentioned in the previous slide. There is a variation between vaginal birth as well as in the c-section, skin contact and breastfeeding also, there will be alteration in the in the gut microbe, the environmental exposure, the diet transition. So the general external exposure will be climate, socio-economic factors, stress, education. So all these are the factors, the common factors. The internal will be metabolic factors, oxidative stress, gut microbes, inflammations, hormones. So externally, the climate also, in a specific region, there will be good amount of the bacteria that is present in the externally. There's socio-economic factors. So what socio-economic factors is like, sometimes the malnutrition, or the all kind of nutrition, what kind of food you're taking. If a person is, the rich people, if you take the economic system, so sometimes people can give a good food, right? If a person is economically below the line, so he can't give a good food, so that nutrition also alters the gut system. The stress, it is one of the main reason why there is a dysbiosis, which you will study in the coming slides, happening, which is a main cause for many kind of GI disorders. And the education, because you study, what kind of food you have to have, because of which the gut system changes. If you have good food, okay, if I say, if in what I say, the good food is nothing, the food that is rich in fibers, okay, the food that is good in antioxidant, all these are the good foods, right? So that will alter again the internal factors, the metabolic factors. Okay, so again, the food changes the metabolic factors, the stress what you're taking, the oxidative stress, maybe not the real stress, maybe the oxidative stress, maybe uh, your the oxygen level in your system is low, that is also creates a certain kind of stress. The gut microbes, the inflammations, the hormones, any kind of imbalance in your internal system also alters your gut microbes. Specific external factors like infection, radiation, alcohol, smoking, diet, physical activity, antibiotics and medications, all these are also the specific external factors that will, you know alter the host gut microbial system. Okay, I hope you're clear with this. Next, you will see here what happens with the diet. Okay, you can see the three kind of diet is it: high fat diet, red meat diet, uh, high fiber diet. Okay, so you can just see the two pictures here. Okay, so you can see here diet that is low in animal fat and protein but high in plant fibers. So what happens? So they will have digestible digestible proteins as well as indigestible polysaccharides. Okay, so we will see that in the coming slides. What is this digestible and non-digestible? So there will be, if the undigested will be, it will undergo the process of fermentations. So basically, what you have to very highlighting thing is this. Okay, so now what happens, that is short chain fatty acids. So this is like a, it is like a, it's like a good push for the system, for the gut system. Okay, it is like one of the important factors, that is small chain fatty acids. So remember, in the low fat diet, the end product will be the short chain fatty acids. So it will have propionate, acetate, butyrate. So this will be, it makes your gut happy. This small chain fatty acid makes your gut happy. Because of this, the glucose, there will be like the optimization of the glucose, there will be increased energy expenditure, glucose stimulated, and insulin secretion. So everything will be online, means on the mark. Okay, everything will be where it need to be there. Our system works specifically, means properly, if all this system is gone, I mean it's in line. Okay, however, if your diet is high in animal fat and proteins, okay, but low in plant fibers, so what happens, you can see all these reactions that is happening. So the peptide breakdown, sulforamines, all the amino acids, proteins are made up of amino acids. So all these amino acids are formed. So this is not completely bad, okay, but what I'm saying is, so this will produce a certain kinds of inflammatory, pro-inflammatory markers, or they produce some pro-inflammatory uh, components that we can see. So that what happens, see, they also produces all these organic acids, they produce all this, components like indoles, all these are formed. So that will increase PAMs. Okay, so these are all the inflammatory, the components. So that will increase the blood glucose level, and also the metabolic endotoxemia, means it will produce toxins. So if I come here and show the slides, you can see here once again. So now you understood what happens between the two kind of diet, right? So here, if I take a simple high fiber diet, there will be short chain fatty acid that is produced. So that will act as an anti-inflammation and anti-acid, which is good for the system, for the body. Next, if you see here, high fat diet, red meat diet, here what happens? Long chain fatty acids and all the other derivatives are formed. So you can see the thrombosis, atherosclerosis, inflammation, you know, all these are formed. So along with that, you can see here, I told bifidobacterium, it's a good one, right? So that will be increased. If you see, sometimes some people diagnose in a certain conditions, they see the microbial, they see the gut microbial, what are the components that is present in the gut. So that time they can see the bifidobacterium. If it is increased, means that is a healthy gut. Okay, so you can see all this microbes, you can see all this. So if this is increased, it means a person is having a healthy gut. You can see here, it is decreased, the enterobacteriaceae is increased. Okay, this is increased, the rest everything is decreased. Okay, so there is a variations, there is a alterations. So this is also, we can it is a comparative factor between a person who is having a good diet as well as a bad diet, okay, which will influence the body system and also a certain kind of the disorders. Next, here you see the slide, we'll see some of the role of the microbiomes. Okay, the role of the gut microbiome. So now, after all this, you studied, you understood various roles of the gut microbes. So that is, one is metabolism and weight regulation. So the microbes helps in the metabolism as well as the weight regulations. Okay. So synthesis of essential compounds. Now you know how it forms essential compounds. You saw short chain fatty acids. So all those are the essential compounds. Okay. Next, protection against pathogens. We will see in the coming slides how it protects against the pathogens. Immune system support, like I mentioned previously, immune system, digestion and nutrition absorption, drug metabolism, brain and gut axis. So this is a very important, gut and brain axis. Okay, so now we will see how the digestion begins. Okay, so if I say it has a function in metabolism. But microbiome, it has a function in metabolism. So what does that mean is, so in the gut microbiomes, right? So it will produce, okay, the influence the metabolisms with specific enzymes. So these will produce a certain kind of enzymes. What it will does, it helps in the, you know, if the larger, the different kind of metabolism, either carbohydrate metabolism or protein metabolism, in the various kinds of metabolisms. Okay, so they basically influence in the metabolism by the production of, by the secretion of specific enzymes. So this is how they help in the metabolisms. Okay, so what happens if the digestible food, okay, so if I say the food, so some foods are digestible, right? So some will be undigestible. So what happens, the digestive food, it will undergo the digestion process and it will produce the ATPs, all we know that. So the undigested food, okay, so this will undergo the process called as fermentation. So the process of fermentation, okay, it is the main source of energy in the colon. Okay, it is a main source of energies in the colon. So the fibers, okay, so those are usually, they are resistant to digestion in the small intestine. Usually the digestible components will get digested in the small intestine, but the fibers are not digested, they are resistant for this. Okay, so what happens, this will undergo various kind of fermentation process. So this will then form short chain fatty acids. you know we will see whatever the components that is formed. So we will see. Okay, so now here it will undergo the formation of fermentations. uh okay, fermentation. Hello, am I audible? I just lost my connection. Hello.
Yes, you are. Yes, you are, ma'am.
Okay. Well, so now I was telling the food that is digestible, so that will be absorbed. So the undigested food, it undergoes the formation of fermentation. So that is a process that will, gives a good energy to the colons, right? The fermentation is very intense and it forms scabs. So these scabs basically acetate, propionate, and butyrate. Okay, so this, what it does is, so in the process of the digestions, so this will again increases the growth of bacteria. Okay, so this is how the food what we consume alter the growth of the bacteria. If you have a good food, if you have a fibers, so that will undergo the fermentation, that will form the scabs, because of which the bacterial growth will be increased, and that the bacteria what is increased, that is a good bacteria, right? So this is what happens in the case of the fibers. Okay, so if the food that is having a the low dietary fibers, low dietary fibers. Okay, the food that has a low, the dietary fibers, it will affect the microbial diversity. So what it does, it decreases the microbial diversity. Decrease the microbial diversity. So this is how the food impact the diversity of the gut microbes. Okay, so this is how again the metabolism also is affected. So again, the good bacteria, it helps the digestions. Okay, so that will again, it has all these kind of functions. The metabolism the metabolism and weight regulation, synthesis of the essential compounds, protection against pathogens. So again, it is like a continuous circle. So it will also be helpful in the process of the digestions. Okay, so in the process of fermentation, so if you take some other components of the food, sometimes there are certain kind of food that will be low in fibers, right? So sometimes what happens, these also, sometimes, for example, the low food or with the high low fiber and high fat. So in sometimes the high fat, what it does, it will also decrease the microbial diversity. Along with that, it will produce some of the toxin substances. Okay, so along with the process of digestion, they will produce some of the toxin substances, the end products. So that will affect the system. So you can see here. So you can see some of the hydrogen sulfide, all these end products are produced, which alters the system. Okay, I hope you're clear with this. Next, intestinal homeostasis. When a relationship is very well established between the intestinal microbiota, luminal antigen, and epithelial barrier, the immune privilege develops, leading to tolerogenic state. So, if there is a well established relationship between the intestinal microbiota, okay, the luminal antigen, and the epithelial barrier, so they will have a tolerogenic state. So there will be balance, and the balance, so all these are the inflammatory responses. Okay, so TH is T helper cells. T helper cells. Okay, so there should be always a balance between helper T cells 1 and helper T cells 2. So if the helper, if there is not if there is an imbalance, so the tolerogenic state is interrupted. Okay, see, understand, it's very simple. So if there is a relationship, well established relationship between intestinal microbiota, luminal antigen, and epithelial barrier, there will be tolerogenic state. So there will be, if there is a beneficial bacterium, that will result in the tolerogenic mechanism, because of which the helper T cell 1 and 2, there will be balance between them. So this is usually present in the healthy subject. However, disruption in the gut might, the homeostasis. The homeostasis means the proper homeostasis. There is a balance. So if this balance is disrupted, the healthy bacteria will be replaced by pathogenic bacteria. So there will be perturbance, means the bad bacteria will come into the system. Okay, so because of which there is a dominance of the helper T cell 2. So this results in some various kinds of conditions. Either Th2 dominance, that will be helping, forms allergy. So in the allergies, there will be helper T cell, T helper cells dominance will be present. Or in the IBD, even in the IBD, they see that there is a excessive production of Th2 cells. Okay, Th2 responses also for the leaky guts. The leaky gut. Okay, so they produce increased amount of the mucus. Okay, which alters the integrity of the epithelial barrier. So epithelium will be there. So all this epithelial barrier will be interrupted. So because of this leaky gut means what? There will be leak, right? So because of which the toxins might go to the blood, or the microbes might come into the system, into the gut, the bad microbes. Okay, again there will be the alteration of system and formations of the various GI disorders. So next we will see what happens, what is the process. Okay, so now, till now I just want to tell you what we did till now. So we studied about the gut microbiome, right? We saw the various diverse microbiomes that are present in the gut, right? From in the small intestine, in the large intestine, all the gut microbes what are present, we have studied, right? Okay. Next, we saw some of the roles of the gut microbiome. And we saw how the gut microbe varies with the diet. With the diet, right? With the diet. Also, we saw how it started from the birth. The microbiome alteration with the birth, right? How happened at the time of the birth, during the development, during the process of feeding, and the adult diet, and the factors influencing them. I hope you're clear with this. Next, we will see the alterations. Now, if the alteration happens. So the first primary word, okay, the term that you have to remember with the alteration of the gut microbiome is dysbiosis. So what is dysbiosis? Is nothing but alterations of the gut microbial composition. So a normal, you can see the picture here, the normal gut will have a good amount of lactobacillus, bifidobacterium, all these are the good microbes. Okay, so if this is altered, means there is a decreased amount of the production of these good bifidobacterium, or there is the you can't find the bifidobacterium or good lactobacillus in the system, and there is an increased number of other bacteriums, maybe candida, klebsiella, E. coli, all these pathogens, if it is present, then it is called as dysbiosis. Okay, the system is disrupted. You can see the gut lumen. Okay, you can see various markers of the gut dysbiosis. So you can see in the normal system, the gut lumen, you can see there are various microbes and the good amount of microbes. There is a variation, right? So there might be bifidobacterium, there is lactobacillus, the actinobacters, all these are present. The good microbes that is present. Also, there is tight junction proteins that is present, that is zonulin. There is a required amount of the zonulin proteins, because of which you can see the the junctions here, the barriers, the epithelial cells, the intestinal epithelials that are tightly packed, right? So you can see how it is tightly packed. Next, here you see in the dysbiosed gut, here what happened, there will be dysbiosis. There is only one kind of microorganisms, so that might be pathogenic. So either E. coli might be present, any kind of what infection has come on, that kind of bacteria which is not good for the system is present. Okay, so along with that, there is something else also. There is loosening of the tight junction. Okay, so because of the dysbiosis, okay, when the dysbiosis happens, it will results in various disruptions. So that will increase the zonulin. So this zonulin, if this zonulin protein increases, there will be leaky gut, means there will be wide opening. So because of which the microorganism can go out of the system. You can see the blood system. If it comes, what happens? Infections, right? Bacterial translocations. So endotoxic will happen. So systemic response will happen. Inflammations will happen. Inflammatory response will happen. So because of this, various conditions are developed. Okay, I hope you're clear with this. So in the gut lumen, you can see it is tightly packed. Everything is fine. However, when the dysbiosis happens, you can see how the alteration affects the system. Well, so now I also told you that the gut microbiome is linked to the immune system. Okay, so how it is responsible, act as an immune system, also responsible to promote the immune system. So how it does is, so if I take this as a good bacterium, so this is a good bacteria that is required for the body. So what it does, it will display some kind of properties. So basically, first thing, it will recognize and attack opportunistic bacteria. Okay, so when the opportunities, any kind of commensals come, any kind of commensals, if it comes, so it will recognize it and it will attack. Since I told, since the lining, the whatever the system, what it is comes from the birds, right, as well as the development. So you will have the good bacteria system already formed in your linings. So what happens if the bad bacterium comes? It will recognize and it will attack. Okay, so using the various, metabolites. So it will produce certain kinds of metabolites. So along this, the metabolites, one is, I told, the short chain fatty acids. So this short chain fatty acid, like I mentioned, it will promote IgA, the immunoglobulin A. So this will, what happens? This will block, okay, this bacterial adherence to the epithelial cells. Means the if this the external bacteria, if it comes, it will block. Okay, so it will, it will directly affect the bacterial virulence. Okay, which will prevent the bacterial invasion as well as infection. So IgA, it will affect bacterial virulence, which is comes externally because of the dysbiosis of anything or because of the food or any conditions, and will prevent bacterial invasion and infection. Okay, I hope you're cleared with this. So this is how the gut system acts. Next, so the intestinal defense system. So now I mentioned there is a tight junction, right? You can see here what are the defense system that is present. In the gut lumen, the commensal bacteria ensures the pathogenic inhibitions. Okay, so the commensal bacteria, it will ensure the pathogenic inhibitions will be present. In the gut wall, the tight junction prevents the pathogen from the entering the body. Like I mentioned, the tight junction. The healthy microbiota triggers the immune system to produce immunoglobulin A. So this is how the healthy intestinal defense system works because of the presence of the healthy microbes that is present. Next, the gut and the brain axis. So this is the next important characteristics that is present, that is gut and the brain axis. So what is this gut and the brain axis? So it is like a communication between the brain and the intestinal microbiota. Okay, brain, intestinal microbiota, and brain. Okay, so the gut and the brain axis is bidirectional. So the direction, the direction or the communication happens both the way. It is not a single line of the communication. It happens in the both the way. So from the brain, there is a information coming to the intestine. Also from the intestine, there is a communication going back to the brain. Okay, so it involves direct and indirect pathway between cognitive and emotional centers in the brain. So the gut and brain axis complex cost that it has a crosstalks between endocrine, immune system and autonomic nervous system. So what it basically is, oh, okay, let's see here. First, we will see the neurotransmitters. And hormones. So the gut produce various neurotransmitters. Okay, the gut produce various neurotransmitters. So what happens? Any kind of stress also it is produced. Various neurotransmitters it is produced in the gut. Okay, so what happens? This will be going to the body, to the brain. So because of which there is a regulations in the brain. So what happens if, for example, so if the, like I mentioned, the gut produces a certain kind of neurotransmitters. If the gut produces, affects the serotonin level, because of which what happens, there will be regulation in the mood. The mood affects the brain function, the mood effects of the system. So if the gut, it sends the transmitters or the hormones or the signaling molecules for serotonin, serotonin balance. If it, if there is an imbalance, the mood will be affected, right? So in the same way, there is something called as vague nerve. So vague nerve is a 10th cranial nerve. Okay. So this has a direct signal from gut to the brain. So signals from gut will travel up to the vagus nerve of the brain. So you can see here any kind of informations that will be, there will be the exchange of the informations between the gut and the brain because of the vagal nerve. So we can see this vagal nerve, there will be sensory signals, okay. So there will be the sensory signals, if I miss sense, from the the GI tract to brain. So it will be like, if the presence of nutrients is there, if there is activities of the gut microbes, if anything will happen, it will send the signal to the brain, because of which the brain gives certain kind of function. So if you have a good carbo, if you have a ice creams, you might feel, sometimes some people might feel good. So all these are the stress response. Okay, so if person is having a lot of stress, they start to eating. So all these are the kind of the gut and brain axis. Also there is a motor signals. So here it comes from the brain to the gut. So here it is basically for secretion of the digestive enzymes or modulations of the immune systems, all this. Okay, so you have to remember there is a vague nerve that is connecting between the gut and the brain, that will be also the taking part between the gut and brain axis, and helps in the increase all the kind of the functions, mainly the gut functions. Okay, so in the gut, mainly because of the microbes, all the signals have gone to the brain, and from the brain all the other kind of message. So there is a continuous you know crosstalk between the brain as well as gut. So some of the diseases of the GI disorders is because of the if a person is having certain kind of GI disorders, they will be taken to the psychological consultations, because there is a crosstalk between the gut and brain. So that you will study in the coming slides. It will be mentioned in the coming slides. So impact of dysbiosis on the mental health. Basically, dysbiosis, it will alter the gut and brain axis signals. So like I mentioned, inflammation, immune activation will happen. So what happens, the dysbiosis, it will lead to inflammations, right? So because of this molecules, there will be the alteration in the moods. Next, neurotransmitters, the serotonin, the dopamine level will be regulated, it will be irregular. Next, short chain fatty acid, this you know, so you already studied, this will have a wide range of importance here. And again, stress response, so any kind of stress, there will be the crosstalk. So any kind of stress, the internal stress or the mental stress, so everything will be altering the each system. Next, the communication via vagus nerve. So vagus nerve, it will, I told you, it will influence the emotion, mood and behavior. So if there is any you know, impairment in the gut system, the vagal inform the nerve, the signals via that is gone via vagal nerve is not provided as it required, it should be gone, right? So there will be the alteration, so this will how it will impact the system. Next now we will see about the gut microbiota in GI system. So this is all the basics, okay? So this is now we are studying about the GI disorder, because if you doesn't know what is gut system and how it acts, you will not know the pathogenesis of all the disorders what I'm going to teach you right now. I hope you guys are very clear. See, very simple, basic functions will be there, and two basic terminology, the dysbiosis, the alteration of the systems. So alteration of the systems, you know, the linings will be disrupted, because of which there is dysbiosis. The microbial diversity will be altered, and the crosstalk between the gut and brain will be differed. Okay. Next, gut microbiome in the GI disorders. So in the GI disorders, you can see various microorganism I have mentioned here. In the obesity, you can see what kind of microbes have increased and what kind of microbes are very decreased. So if I read, it is, it is very difficult for you to recognize, but you guys have to remember. So there are certain microbes which is good for us in a good amount, and there are certain microbes that is not good for us if it is present in a excess amount. So you can see how it varies in certain metabolic disease, see obesity, type 2, because all these are prone to in the future some other kind of you know the related disorder. So it might affect from, it might start from obesity, then it might go to NAFLD, T2DM, any kind of disorders. So you can see various microbial arrangements. So now we will focus on the GI disorders. So first it is irritable bowel syndrome. So irritable bowel syndrome, there are various subtypes of it. That is IBS-D, that is with diarrhea, IBS-C, that is with constipation. Okay, and there is mixed. So mixed means both. So you can see the characteristics that is present with the IBS. So in IBS-D, the diarrhea will be most common in men. Okay, and IBS-C, the constipations will be most in the females. Okay, so this IBS is nothing but it is a FGID. It is a functional gastrointestinal disorders. Okay, so basically the patients with FGID or IBS, they will have a pain and discomfort. They will have a altered bowel habits. So the prevalence is increasing because of the various, the food habits. The food habits is changing, right? So because of this, there is the prevalence rate also, because it was very high in other, the US, all these countries. Now it is increasing even in the Asian countries as well. Next, pathophysiology of IBS, the gut brain axis. So I hope you're clear with now what this gut, brain axis is. Like I mentioned, it is a bidirectional relationship. It is a bidirectional relationship. Okay, so it is basically, if you say brain, it is consist of the central nervous system, neuroendocrine, and neuroimmune system. System as well as autonomic nervous system, enteric nervous system, and the gut microbiome. So this is the two components, the whole nervous system as well as the gut microbiome. So here what happens, there is a hypothalamic pituitary adrenal axis that plays a key role in the gut signaling pathway. So you have to remember HPA axis. Hypothalamic pituitary adrenal axis. Okay. So now what happens? So this, it will be in the homeostasis. So there will be, this axis is basically a neuroendocrine system that helps in the maintenance of a homeostasis through several cascades. So what happens if there is, this homeostasis is disrupted? So it is because of the various stimuli. So one stimuli, let's just take as a stress. So what happens? The stress, what happens? The stress, it will, basically this try to it will maintain the homeostasis of the system. So now what happens in a stress condition? What happens? The hypothalamus releases corticotropin releasing hormone. Corticotropin releasing hormone. Okay. So what this hormone does is, it will subsequently prompt the anterior pituitary to release adenocorticotropic hormone. Adenocorticotropic hormone. Understood? If there is any stress, you can see this diagram here. If any kind of the emotional stress or any kind of stress is present, it will produce the hypothalamus. Okay, the hypothalamus will produce corticotropic releasing hormones. What it will do? It will subsequently prompt the anterior pituitary to release adenocorticotropic hormone. Okay, so this adenocorticotropic hormone, it will go to the adrenal gland. So this stimulus, okay, it goes to the adrenal gland to release cortisol. Okay, so this is body's main stress hormone. So what happens? This will go to the intestine. So what this it will do? This will also act as a pro-inflammatory, stress hormone. So what happens? Because of this, there will be pro-inflammatory markers will be increased. Interleukin 6, interleukin 8, also the neurotransmitters will be increased. So the activation of all the cellular effectors can lead to the dysregulation of HPA axis. Okay, so what happens? There will be increased inflammatory markers, increased cytokinins. There will be, increased hormones, norepinephrine, serotonin. So because of all this, all this process, it will be dysregulated, right? So activation of this all cellular effectors can lead to the dysregulation of HPA axis. So this is one of the, it is a pathogenesis of IBS, because of which the IBS is formed. So the high level of the psychiatric consult is given. So if a person's psychiatric, condition is balanced, then also his IBS situation is also maintained. Okay, so this is about the pathogenesis of the IBS. So what happens? It also happens not only because of the stress, because of the dysbiosis. So dysbiosis also sends certain kinds of messages to the brain. Okay, so you can see the low grade inflammation. See, cellular and mucosal immune activation. So again, that will produce low grade inflammation system. So that will also alter the HPA axis. So basically, this you guys have studied right now: changing gut mortality, alteration in the nervous system, all these. Okay. Next, another condition is SIBO. So SIBO is nothing but small intestinal bacterial overgrowth. is another clinical conditions, okay, characterized by gut dysbiosis. So here what happens, because of the dysbiosis, there will be excess of bacteria in the small intestine. So because of which there will be fermentation happening, okay, the fermentation of the indigested carbohydrates happening in the gut. So this will be leading to the production of the gas, okay. So if a patient comes to the production of high amount of gas, or abdominal pain, bloating, all these are the conditions of the SIBO. Means they have a externally, they have a no, they have a extensive bacterial growth in their uh, small intestines. Okay. Next is inflammatory bowel disease. So first you studied irritable bowel syndrome. Now is inflammatory bowel disease. So in inflammatory bowel disease, like the name it says, it is about inflammation, right? So like now, I hope you guys know how the inflammation comes up. So if there is dysbiosis happens, that will give rise to inflammation. Okay, because of what happens, the microbial dysbiosis happens. So the microbials comes here and there, right? See, the good microbes changes. If the pathogens comes, what happens, our immune system will be activated, right? So the activations of the immune system leads to what, the cytokines, all the immune factors will be increased. You can see here TNF alpha, all the interleukins, all these kinds of the inflammatory markers are increased, because of which there will be production of the inflammatory states. This will result in the inflammations in the colon regions on the intestine. Okay, so you can see here some of the bacterium that is increased in the IBD and that is decreased in the IBD. So these are some of the bacteria: Fusobacterium, Pasteurellis. So all these are the bacteria or the fungi groups that is increased in IBD, and these are the bacteria and fungal groups that are decreased in the IBD conditions. So this is again same, like I mentioned, if there is a good microbiome, it displays a anti-inflammatory effects. If there is a bad microbes, means which is not good for the system, which can be because of the antibiotics, diet, breastfeeding. I told you, the breastfeeding, if you know it, because of any kind of breastfeeding, if the person is having any kind of antibiotics, if he's not getting a good amount, so the back system, the environmental factors, so also the genetic factors, so it will give rise to the pro-inflammatories. So this will, what happens, this will, there is a dysfunction in the barrier, the epithelium is there is a dysfunction, the bacterial translocation happens, the immune system activation happens, and all the immune cells are activated, which is results in the IBD. So in the individual system, commensals bacteria provide functions that benefits the host. Okay, how certain bacteria produces, that is small chain fatty acid such as butyrate to regulate intestinal homeostasis. So antimicrobial peptides produced by all these cells, okay, intact mucuses prevents the bacterial invasions of the epithelial cells. So this helps in the maintenance of the functional barrier, also provide anti-inflammatory properties, okay, by facilitating the differentiation of the T cells and affected uh, regulatory T cells and effective T cells, right? So next, what happens in patients with IBD? There is reduction, so including the SCFA, the small chain fatty acid producing bacteria. So this will trigger the cascades, okay? So because of which there is the bad bacteria has the opportunity to emerge, also form the inflammatory conditions. So in the IBD, the risk factors will be not only microbes, even the genetics also plays a factors. So this is all the risk factors of the IBD. Next, microbiome and systemic diseases. So systemic diseases may be obesity, type 2 diabetes. Okay, so we can see here, because of the two things here, mechanism by which intestinal microbiota are associated with obesity and diabetes. So you can see here, short chain fatty acid and gut permeability. So short chain, I think by now you know how it works. Gut permeability also. So what happens? The disruption of the gut barrier function and the gut microbe divide endotoxinemia could contribute to the pathogenesis of the vitamin, the obesity and T2DM. So here one of the studies, okay, I've mentioned this slides will be shared, the recordings will be shared. So you can see scientists have conducted one of the studies, okay. So what they did is they took a mice that was high fat diet mice, and they saw some of the characteristics of the mice. So when they studied the system of the mice, they saw that the gut system, it had an increased gut permeability. So the gut will was not, it was not holding very tight, the permeability was more. So because of which what happens, the barrier function is the disrupted, because of which there is a translocation of the bacteria, also modulation of the bacteria, the modulation of microbiota by antibiotics, okay. Okay, so what happened? So they took a high fed you know fat diet mice with the increased gut permeability, also modulation of microbiota by antibiotics, okay. Understood? So increased gut permeability. So you can see here leakage of lipos polysaccharide into the portal circulation. So what happens? So because of the lipopolysaccharides, there will be storage of the fat in the system, which might cause atherosclerosis. So here what happened? The same mice, later what they did a modulation. So how they modulated? By antibiotics or prebiotics, because of which there is reduced metabolic endotoxemia, lowered inflammations, and elevated glucose intolerance. You can see here. So once the mouse was modulated, there was a reduced metabolic endotoxemia. You can see what happened to the system. So you can see the effect of the the fat as well as the treatment. So both the you can see here. So microbiome and liver disease. So I just thought I'll put some slides on this. The same functions. If the inflammation happens, so inflammations, like I told, the chronic inflammation, that will change the system. So chronic inflammation is nothing but the what happens? It will starts with the fibrosis, then it will lead to cirrhosis, right? So you can see as CAS, it will produce butyrate. So here the intestinal permeability increases. If that is not happening, it will activate the immune system. The inflammation will increase, because of which there is a result in the liver disease. So not just this, there are many other factors, okay. This is one of the factors. So ethanol also you can see here, cross activation happens. So this is one of the internal stress, because of which also the gut microbial system alters. The same thing happens here. PAM, bacteria, all these are the inflammatory factors. You have to remember, inflammation is a triggerer here. So in this table, you can see some of the studies of the gut microbes in the NAFLD patients. So NAFLD is non-alcoholic liver disease, right? So you can see here the studies that have happened in various US countries. You can see here the NAFLD enriched taxa. So all these can act as a enriched taxa. Means if in the future, if you're going to study, if these microbes are more, so that person is having a NAFLD or prone to get a NAFLD. So microbes and GI cancer. So here you can see in esophageal cancer. So these are still in the learning phase. So this I've taken from some of the recent studies that have happened. So in esophageal cancer, it is reported that Streptococcus viridin is one of the most common microbes that is present. So here the transition from the gram positive bacteria to gram negative predominance in the esophageal flora is the important inflammatory response, okay. Basically the inflammatory response is the one that will trigger the system and causes inflammation, because of which there is a alteration in the system. In the, gastric cancer, it is very popular, that is Helicobacter pylori. So Helicobacter pylori, pylori is the best known example for causing gastric cancer, right? So this pylori, it injects cytotoxins, VAC-A and CAG-A, okay, vancomycin. So what it does, so it activates the oncogenic signals for activations of various carcinogenic pathways. So it is very popular that gastric cancer is, Helicobacter is one of the reasons for the causative for the gastric cancer. Next, colorectal cancer. Here again, E. coli also plays various, you know, it is, studies have gone that it could be the reasons for, the formation of the E. coli. So, but along with that, these are still in the study stage, and many other derivatives also are present. Okay, so they are saying that microorganism also can drive the colorectal cancers. Okay, so you can see here CRC, that is colorectal cancer associated bacteria. You can see what are linked. All this. So this is, in the few studies they have shown that these many microorganisms are present in the abundance in the fecal and the tumor samples from a CRC individual. So these are, I told you, all these are still in the learning phase. Okay, there are a lot of researches happening in this phase. The microbes, because this is a new phase, I told you that is a new next generation treatment. So recently they've learned us, found one of the microorganisms which can also trigger to produce insulin in the system. So there are a lot of studies is happening. This is very important. Intestinal microbiota modulation as a novel therapeutic strategies. Okay, so now you understood what dysbiosis causes, what are the various kinds of, GI disorders dysbiosis causes. So now let's see what are the modulation we have to do to whatever, cure whatever happened. The modulations, okay, as a novel therapeutic strategies. So basically, untargeted intervention. So untargeted will be exercises, individual nutritions, fecal microbiota transplant. Targeted will be bioengineered commensals, any kinds of the drugs, okay, or CRISPR based or phage therapies. All these are very specific, very targeted, which is still in the clinical phase. Okay, there are many things happening in the clinical phase, or in the in vivo they are trying this. Okay, this you guys have studied what happens with the diet. So I told you the untargeted, the nutrition, the diet. So how it, already how the diet, the diet, alters the gut system. The dietary fibers, short chain fatty acid will be produced, which is good for the system, and it will also increase the gut microbes. So high fat diet, so it will be increasing the gut permeability. It will causes the dysbiosis. All this will happen. Next, the very important, the prebiotic, or the it's very fancy now. Everybody is having prebiotics or probiotics, all this. So what is this? So these are the basically two things, that is the symbiotics, that is contains a mixture of prebiotics as well as probiotics. Okay, so basically these are nothing but the modified live organisms. It can be a food, or it act as a component. So probiotics are the live bacteria. Okay, it is a live bacteria. So it is a live bacteria. Okay, when administered in a viable form, it is in an adequate amount, it is good for the human health. So it is a live bacteria. So here they are like usually yogurts or supplements. Prebiotics are the substrate. Okay, this is direct component. Here the direct component, but here this is a substrate. Some substrate will make the microorganism grow. Okay, this is the direct the live bacteria or the yeast, all this. Okay, so this is like in a particular kind of carbohydrates or fermentable dietary fibers. Okay, so that is make the microorganisms, particular microorganisms. So for example, if you want bifidobacterium, so they will target a particular substrate for that. You know the studies, because scientists do continuous research, they will know what kind of substrate increases bifidobacterium. So this is why people say you have to have good curd, all this, because it will carry a good amount of, it might be the substrate or the curd, it will carry the bacteria as a good substance. Okay, so probiotics are helping in the treatment of all many kind of GI disorders. You can see here in the picture, in the diarrhea, in the infection, lactose intolerance, IBS, IBD, as well as H. pylori eradications. Okay, so now in the previous slide, this slide, I am repeating this slide, you understood whatever happens. So what happens if the defense system is breached? What happens? Defense system is the breach means dysbiosis. So if the dysbiosis what happens, the pathogenic bacteria dominates, right? The pathogenic bacteria will be dominated. Also there will be translocation because the gut wall is not tight enough. Also the immune system is not triggered, the required immune systems. Okay, so probiotics, what it does, it will compete for the nutrition. Okay, the probiotic tighten the cell junction and enhance the gut microbes because it is having a good microbes in them. The probiotics also trigger the immune systems produce means IgA, not the inflammatory, the immune systems will be triggered and neutralizes the pathogens. Okay, so another system is fecal microbiota transplant. So what they do, they take the fecal samples. Okay, so from the fecal samples they take the required the microorganisms. So that microorganisms, it will be then like it form you form a the tablets out of it and they are given, or directly it is given to the systems. So FMT is reported to be highly successful treatment for Clostridium difficile infections. Okay, so again, this is the application is still ongoing and the treatment is known for IBS, IBD and certain GI disorders. So again, the probiotic uses, you can see here various IBS, IBD, Bifidobacterium infantis for IBS. So E. coli, certain strains of E. coli, not all the strains of E. coli. Okay, for IBD, you can see here these are the very strains so that are present in the probiotics that are taken. So it is happening, it is running in the market right now. So there are various companies that is providing the probiotics tablets that will have all these components which will increase the gut health. So basically, in this system, you have studied about the gut microbiome, you have studied the various disorders, also its implications. So if I have to say the targeted treatment, the bioengineered system or the drugs, so all these are the bioengineers very specific. The probiotics all will be very specific. Okay, when it comes to targeted phage therapy, phage. Okay, it's a virus that will attack the bacteria, right? So if we doesn't want a particular kind of bacteria that is not supposed to grow, we will give the phage treatment. Okay, this virus will go and kill those bacteria. So CRISPR-Cas, this is again still in the clinical step, preclinical steps. All studies are still going on. So here they will modulate the bacteria's DNA. The gene is modified. They make the bacteria to produce good epithelium barrier, or the bacteria that will, I told you, insulin if it is there in the maintenance of T2DM, or any kind of enzyme production, good enzyme productions. So they will alter the system. So this is very targeted. So this is about the gut system, the gut microbes, and the related GI disorders. So you guys can ask any kind of questions. Yes, yes. Gut microbes can reverse. So that is where the probiotics, all this, the rules can take place, right? Even the good food, it can alter the gut microbiome. Okay, is the leaky gut reversible? See, leaky gut reversible, it will goes up to the, the point of the extent of the damage that happened to the system. See what happens. See, leaky gut is not the problem. So leaky gut is a primary problem, okay, of some kind of big problems. For example, if a person, so I told in the IBD, a leaky gut can happen. So once a person has developed the inflammation in his gut, correct, the inflammatory of bowel, so what happens, that is a permanent problem, right? The inflammation, IBD is formed. Or because of the leaky gut, the leaky gut, if a person is having, the inflammatory components is formed, and then the liver fibrosis happens, then the liver is damaged, right? So this is about the reversible factors. If it is found in an early stage, it can be reversible because of the food habits, because of the diet, because of the exercise, because of the, all these probiotics, therapeutics also, as well as the food and the nutrition. So that is what I'm saying. So this is a future of the medicine, okay? Because we are coming back to the age old medicine where we started saying, because in the previously what it was told, food is a medicine, right? So we are coming back to the same phase. We are even now, it is, the studies have come up, it is showing, see, this microorganism is good. So what happens if you eat a certain kind of food, you know having to produce a certain kind of bacteria. For example, if I say spinach, it will produce a particular kind of bacteria. So it means what, you will find some kind of nutrition, right? This is why some food is good for a person, okay? Okay, is there any other question? So there are, see, presently there are lot of studies happening even in the South Indian hospitals. No, there are a lot of studies are happening based on the prebiotics, probiotics. Okay, so basically they are studying the gut. So basically you have to study the gut. See, sometimes what happens, the prebiotics that is coming from the US company, that they will not work on your system. Okay, very careful. So if a big ad comes, what is, in the abroad countries, okay, this probiotics, it will not work, because they have designed the probiotics based on their systems. The the Western diet is very different compared to the Indian diet, right? So it is, I prefer, like, if you are taking a good food, no, it should be always from your background, like your uh, the specific for the region, also your the probiotics that was developed from the Indian settings, okay? So any other questions? Okay, if there are no other question, I think we can wind up the session.
Yes, ma'am. There is no more questions. We can wind up.
Okay. 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 Which do you think is better, probiotics or prebiotics? Okay. Well, so see, basically now there are a lot of probiotics in the company. So prebiotics is nothing but the food what you have. Okay. So that will be the prone to the body. So I, like, if your diet is good, that is a good prebiotic for the, bacteria that is present in your gut. So it is like a balance, okay? Probiotics is what you take from the outside. So if your food is good, I don't think, I think food is a better option. So whatever you're taking means what are the substrate you're giving for your bacteria, the gut microbe is better than the uh, externally taking something. Okay, the certificates will be shared soon.
Certificates you will get by tomorrow evening probably. And right now we are sharing the feedback form in the group, so kindly fill it. Also, don't leave the group, so we will share with you in the group only entire things. So be in the group.
Okay, can you give a short overview of the things again? Okay, well, um so basically it's about the, like I mentioned, it's about the gut microbiome. So basically we saw various the diverse microbes that is present in the gut. Also we saw how the gut microbiome has evolved in a one system from the birth. The way of birth also is effectful in the development of the gut microbiome. Also the food habits, also from the breastfeeding to the food habit to the complete adult diet. So we saw what are the diverse microbiomes, also the variations among the stage of the development of a, okay, one second, okay, stage of the development of the one human, right? So after that we saw what the dysbiosis is, this alteration in the gut microbe, what it results in the dysbiosis. We saw the gut and brain axis. After that we saw various GI disorders, how GI disorder happens because of all the dysbiosis or all this effects, all this irregularity in this gut system, or all the translocations that is happening in the microbiomes of the intestine. Okay, so there is next question. Okay, how does the postbiotics have a beneficial and harmful effects on the, you know? So I think the postbiotics is coming. It's a kind of ongoing research. Okay, so currently I cannot say whether it really has a good, it is really being a beneficial or it is a harmful. So a lot of studies requires to justify the statement. However, so sometimes some kinds of enzyme taking externally might be useful for a certain kind of conditions, if that condition have reached a certain level. But if a person is healthy enough and is experiencing a very slight changes, or a healthy person, if he have to start, now you have gained a lot of knowledge, so you can just go with a good food, right? And a good prebiotics, probiotics, or probiotics, right? Okay, what are the different bacteria? Basically 16s rRNA they do, this 16s rRNA PCR test, all these they do. They also do RT-PCRs to see whether there are organisms for viral, if some kind of viral microbes that is present in the GI. Basically 16s rRNA, any kind of microbes, they see 16s rRNA, and they do the sequencing also. The recordings will be shared soon. Well, see again about this probiotics. Okay, see, the probiotics is helpful, well, but how much helpful? See, there are the recently, okay, there was a study on hepatic encephalopathy and the use of this probiotics. So they've not seen a much difference. Very few studies have shown some of the functions. So again, what are the the results, that is also important. uh, I feel it is better to have a good diet. Okay, good a diet is better than everything. So probiotics can be consumed, but not on a regular basis, maybe. Okay, so post antibiotic and post biotics effects seems, I'm not very sure about this concept. Maybe uh, I can share if there is any studies happening with this in the group. Okay, I can clarify that in some time. Yes, recordings will be shared. We can even share the PPTs. I can just can do that. Okay. So again, once again, there is a guy a probiotic. Okay, ma'am, if your guy intake probiotic regular, does it cause it? Yes. See, like I mentioned, it's better you take a good food, good diet. So regularly, it's not preferable because it can cause some kind of alteration in the system because you're feeding it in a regular basis. Because at the end of the day, it is something externally is given, right? It is some kind of a supplement, right? So it can cause some kind of the GI you know malfunctions, but very not a huge, but very in a slight manner. Okay, is there any other question? Okay, then I think we can wind up with the session.
Yes, ma'am, we can mind. Thank you
Okay,
so much, ma'am.
okay, thank you everyone for your time and patience.